Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of the year 2018. If you are new to my channel, please do like, share and subscribe. Coming to the year June 2018, a uracil containing plasmid was constructed and was used in transformation into wild type UNG plus and uracil and glycosylate mutated UNG minus E. coli cells and scored for transformants in the presence of appropriate antibiotics. Which case of the following statement correctly describes the experimental outcome? So, uracil and glycosylase, it removes uracil and adds thymine. So in the E. coli cells with UNG plus, when they are transformed, the plasmid with uracil will be repaired and made into plasmid with T. So you will lose your original plasmid. So the number of transformants will be less in the case of E. coli UNG plus. In the case of E. coli UNG minus, the transformants will be more because plasmid will be in the original state. So the answer here will be UNG plus cells will have fewer transformants compared to UNG plus minus cells. Answer is option 1. Then next one. E. coli takes 40 minutes to duplicate its genome using a bidirectional mode of replication. If E. coli were to use unidirectional mode of replication to synthesize a full copy of DNA complementary to just one of the strands of the genome, it would take. So it will require double the time, so it will be 80 minutes. Answer is option 2. Then next one. Transcriptional regulation of TRP operon by tryptophan involves binding of tryptophan 2. So the repressor protein and inhibition of transcription by its interaction with the operator region. Answer is option 1. Then next one. Phosphorylation of EIF2 alpha subunits leads to. So once phosphorylated, EIF2 shows increased affinity for EIF2B. So the answer will be sequestration of EIF2B because of tight binding between EIF2 alpha and EIF2 beta. Answer is option 2. Then coming to our part C question. Actinomycin D inhibits the process of transcription in both prokaryotic and eukaryotic organisms. The following statements are made about actinomycin D mediated inhibition of transcription. Actinomycin D inhibits transcription from a double-stranded DNA template by either E. coli or yeast RNA polymerase. That is true. Then next one. Actinomycin D inhibits transcription from a single-stranded RNA template by eukaryotic viral RNA polymerases. It inhibits double-stranded DNA, not single-stranded RNA. So B is not correct. Actinomycin D inhibits transcription from single-stranded Cy-X174 DNA template of E. coli RNA polymerase immediately after viral DNA entry. So Cy-X174, it is single-stranded DNA, not double-stranded DNA. So it inhibits double-stranded DNA, not single-stranded DNA. So C is also not correct. Actinomycin D, it inhibits transcription from double-stranded RNA template by eukaryotic RNA polymerase 2. It does not bind to single-stranded RNA or double-stranded RNA. It binds only to double-stranded DNA. So A only is the right answer. Actinomycin D inhibits transcription from a double-stranded DNA template by either E. coli or yeast RNA polymerase. Then next one. During maturation process of some RNA molecules, formation of a 2' 5' phosphodiester bond take place. So 2' 5' phosphodiester bond take place in lariat formation. Following statements are made about the phenomenon. Which one of the following combination of the statement is a true representation? So spliceosome mediated removal of intronic sequences occurs through the formation of a 2' 5' phosphodiester bond. That is correct. Removal of group 2 introns occurs through the formation of 2' 5' phosphodiester bond. That is also correct. Enzymatic removal of introns from the yeast tRNA precursors involves 2' 3' phosphodiester bond formation. So tRNA processing, it is mediated by RNase P, and RNase P is not involved in lariat formation. RNase P mediated 5' end maturation of tRNA precursors involves formation of a 2' 5' phosphodiester bond. That is also not correct. So the answer here is option 3, A and B only. Next question. During translation in prokaryotes, when ribosomes reach the termination codon is recognized by the class 1 release factors, leading to the release of the polypeptide. A second class 2 release factors facilitate the termination process. Which of the following statement regarding the mechanism of action of the release factors is incorrect? Class 1 release factors decode the stop codon, while the RF3 is a GTPase that stimulates recycling of the class 1 release factors. That is correct. They have asked for the incorrect statement. Free RF3 has higher affinity for GTP than GDP. It is a GTPase, so it will have more affinity towards GDP. So option 2 is incorrect statement, and that is the answer. Then next one. E. coli DNA ligase catalyzes formation of a phosphodiester bond between the adjoining 3' hydroxyl and the 5' phosphoryl ends in DNA duplexes. The energetic need for the reaction is met by the hydrolysis of NAD to NMN plus and AMP in a 3-step reaction. Following statements are being made about the mechanism of this reaction. Based on the statements made above, identify the correct sequence of the reaction steps. So first will be statement 2, that is adenyl group of NAD plus is transferred to the epsilon amino group of lysine in DNA ligase in the form of phosphamide adduct. The next statement will be option statement 1. AMP is linked to the 5' phosphoryl end of the nicked DNA. The next will be the third statement. DNA ligase catalyzes the formation of a phosphodiester bond by the nucleophilic attack of the 3' hydroxyl group onto the phosphate and releases AMP. Answer here is option 3, that is 2, 1 and 3. Coming to the year December 2018. Which one of the following activities is not involved in protein folding in the endoplasmic reticulum? It is protein ubiquitination. Answer is option 4. Which one of the following statement is not correct? Together with proteins, rRNA provides a site for polypeptide synthesis. Yes, that is correct. All DNA molecules are unbranched polymers of nucleotides. That is also correct. DNA is synthesized in a 5' 3' direction, while RNA synthesis occurs in 3' 5' direction. That is not correct. RNA synthesis also occurs in 5' 3' direction. A tRNA anticodon may pair with more than one codon. Yes, that is also correct. The next one. What would be the tripeptide produced by translation of the transcript produced by the following DNA sequence? So the RNA will be complementary to the sequence. So it will be UUC, AUG, AGA. UUC, AUG, AGA. So if you don't know the first and the last codons, the central one, that is AUG, it codes for methionine and it is present only in one of the options, that is 4. Phenylalanine, methionine, arginine. So answer will be option 4. Then next one. Which of the following statement is generally true about RNA polymerase II? It is it is dedicated to transcribing RNA from a single transcription unit, generally large transcript, which is then processed to yield 3 types of ribosomal RNA. Not ribosomal RNA, it is mRNA synthesis. RNA polymerase II is involved in mRNA synthesis, not ribosomal RNA synthesis. So that is not correct. Then next one. It transcribes varieties of small non-coding RNAs which are expressed in all cell types. That is also not correct. It generally synthesizes various types of mRNA and small non-coding RNAs. That is correct. It is exclusively involved in the synthesis of rRNA and tRNA. rRNA is by polymerase I and tRNA is by polymerase III. So that statement is also not. Now coming to part C questions. Presence of selenocysteine in proteins in E. coli is the consequence of. Answer is option 3, that is amino acylation of a special tRNA, that is tRNA by serine tRNA synthetase with serine, followed by further modification of the attached serine to selenocysteine, followed by its transport to the ribosome by cell B. Then next one. An intron in a yeast reporter gene carries a mutation in the splice site branch point. To suppress the mutation, a library of point mutants of snRNAs was introduced into the mutant strain. The suppressor is most likely to have a point mutation in. So it is U2 snRNA. Answer is option 2.